So translation. Now, as I've watched some videos, the way one of the narrators kept transcription and translation apart, we know the production of RNA comes before the production of protein. Well, she said the C in transcription comes before the L in translation, and that's how she keeps switch apart. I just like our transcript translate alphabet. So what we're doing with translation is we are changing our language from nucleotides to amino acids. But it will be the nucleotides and the codes that are present in those three-letter words called codons that will dictate and direct which amino acids go in which sequence. Now, transcription. Relative to the cell, where does transcription occur? Where does transcription occur in the cell? In the nucleus. What is the product of transcription? And specifically, the one we're going to focus on is mRNA, messenger RNA. So the messenger RNA has to leave the nucleus so that translation can occur where? In the cytoplasm. That's the purpose of the message, because we're not going to get the DNA out, it's too big. We don't want to break down the nucleus because we're not undergoing cell division. So the copy of the code as a message leaves the nucleus and it goes out into the cytoplasm where we can begin to assemble all the components we need for this final step of protein synthesis called translation. So we have our messenger RNA, that's the code. It's identical to the coding strand of DNA with two exceptions really. What are the two differences between our messenger RNA and our DNA? Thymine in DNA, uracil in RNA. What's the other distinction between our DNA and RNA? Single-stranded RNA, double-stranded DNA. So our mRNA has the code. We're also going to require two other types of RNA. We have transfer RNA molecules. Whereas messenger RNA is the instruction, the transfer RNA are going to be our factory workers that go grab an amino acid in the cytoplasm and bring it to the factory, which is going to be our ribosomes. And the transfer RNAs are going to be able to recognize specific codons so that they then know in what order to assemble the amino acids inside of our ribosome. Now, a ribosome is going to consist of a large and small subunit. And these subunits are comprised of proteins and ribosomal RNA. So our three types of RNA are mRNA, tRNA, and rRNA. And these are the three essential components we have to have if protein synthesis is going to be completed in translation. We have other players, and when we get into the specifics of those, we'll add the detail. But right now, we're looking at these three major players in the process of translation. So essentially, we have three steps, not terribly different than we saw when we were looking at our prokaryotic cells and transcription itself. We had the initiation, we had the elongation, and the termination. You got to know where to start, you got to extend the protein chain, the assembly of our amino acids, and we have to know where to stop the process. So this is our overview of translation. And I found a really interesting little video clip that I want to see if I can get to play right now that gets into this overview with a little bit more detail. And I think it will help us understand these processes as we begin to really piece out those finer order details. So this is an overview of translation.
Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
We'll rewind this so you can watch it. And the ribosome is like reading along the mRNA. And as it does, it's sliding the transfer RNA from this first pocket to the second to the third as the ribosome's moving. So it's like an old-fashioned typewriter moving along the paper. So this transfer RNA that has the amino acid, it just came in. Here's the next one getting ready to come in and land. So this is called the A site for the arrival. That's the way I think of it. This is the arrival site bringing in the new one. This is the P site for the growing peptide chain. And this is the E site. And guess what E stands for? Exit, because it's getting kicked out. See, that one got kicked out.
So then when they leave, where do they go?
They just float around until they find another amino acid to pick up and bring it back. They're just shuttling amino acids back and forth.
So how do they know to fly in?
Random collisions. I mean, you got a bunch of these transfer RNAs bound to their amino acids, and they're all bumped.
Long time.
Well, in our guesstimation of a long time looking at this animation, but understand this happens, I mean, extremely fast because of all the collisions and how much the concentration of the transfer RNAs with the amino acids. So it happens really rapidly.
They don't have like chemical attraction?
Well, the chemical attraction is going to be the match. But you know, you're going to have some bumping in. It's going to be like, okay, well, that's not a match, that's not a match, that's not a match. Until you finally get that right anti-codon match, and boom, it sticks. Now, as the ribosome is reading along, this is the key part too. As this empty transfer RNA in the E site gets ejected and makes space, the ribosome slides, and as it does, this middle transfer RNA, it exchanges the growing peptide chain that forms a peptide bond between these two amino acids. Once that peptide bond is formed, the amino acid comes off of the second one and it sticks to this one that slides into the P site, making the A site available for the next one to come in and again and again and again. Does that make sense?
So we've got our A site, which is our
The arrival.
arrival.
Uh-huh.
We've got our P site, which is our
The peptide chain, you're going to form the peptide bond, correct?
exit.
And the exit, it gets kicked out. And that's going to happen, it's just going to, and that chain will grow. The two things I also want to mention, here, this protein is just floating out in the cytoplasm, so this is a free ribosome. It's making a cytoplasmic protein. But if that ribosome were on an endoplasmic reticulum, as a rough endoplasmic reticulum, that protein, as it's being made, it's getting extruded into the lumen of the endoplasmic reticulum. If it completely goes in, it's going to be a secreted protein. If it goes in and a part of it gets stuck in the membrane, then it's going to be a transmembrane protein, maybe on the plasma membrane or in the mitochondria. You see how that works? Now, if this is a cytoplasmic protein, and it's got a special sequence on it that says I need to go to the nucleus, which is that nuclear localization signal, that's going to be the piece of that protein that binds to important that gets it transported into the nucleus. All right? So that's a little bit of the difference, and you'll see this come up in the rest of the lecture between just free ribosomes in the cytoplasm versus what happens in our endoplasmic reticulum. So now that we see the overview, hopefully this will make a little bit better sense as we step through this. Because here are our players, messenger RNA, there's our AUG. We've got our small and large ribosomal subunits. There's our transfer RNA with amino acid number 1 And what did we say our anticodon here was for methionine on the board? UAC. What do we have right there? UAC, amino acid number 1, is always and forever going to be which amino acid? Methionine, because AUG is the start codon. They even painted it in green. There's 3 of them. There's 3 of them. But which one do I remember? How do I say it? What's the gene? The DNA sequence. TAG, tag, like freeze tag, to stop. But you're right, in the RNA it's UAG. But the TAG just helps me remember freeze tag, stop. But then there are 2 others as well. All right? But in this case, there's our UAG stop codon that they show in red.
So for the test, we need to know all the codons?
And again, that last chart that I made, which was sort of the combination consolidated, hopefully that will make it a much easier task. Because remember, there are only 3 that had 2 different ones. So for all the rest, there was one set of codons, either on the end had the R or the Y or the H, representing the UC or the G.
You have that chart on the one that you put on YouTube.
It's in the podcast. I haven't uploaded the new PowerPoint yet, and I'm sorry, I forgot to do that. But it's in that podcast. If you pull up the podcast, yeah, but I'll get the PowerPoint uploaded. So as we assemble our players, the first stage is that initiation stage. And this is all going to be assembled when we bind our transfer RNA that contains the methionine to our messenger RNA. We're going to assemble the ribosome around that starting point. So this is going to be the first messenger RNA that's going to come in before the ribosome is completely assembled. You start with a small subunit, it binds to the message, transfer RNA, and then you put the cap on it. And then once you have this assembled, now we can elongate. Just like in that video, we bring in our A site transfer RNA, we shift over the amino acid to our growing peptide chain, slide it over. There is our, we'll say that's the transfer RNA for methionine. It's gone. And you keep doing it and doing it and doing it until we get to the stop codon, where we're actually going to have something that resembles a transfer RNA, but it's not. It has an anticodon. That anticodon recognizes our stop codon, but it doesn't have an amino acid and it has a different shape. And so when the ribosome moves into place and you see that release factor in the A site, it's like a stick of dynamite. Boom, it blows this whole thing up into our individual components again, but the completed protein is free to do what it's going to do. So again, more of an overview, adding more details as we go. Any questions so far? Okay.
Sorry, so the last part, is that the exit site?
This right here, that's the E site. So A, P, and E. Ape. Makes a word. So if you remember apes, you can get those sites in order, although it goes from, yeah. Really here, in explaining our ribosomes, you got a big one and a little one. They call them 50S and 30S, but for mammalians, humans, eukaryotes, 60S and 40S. The S stands for like a sedimentation factor, because you can separate these with centrifugation. Remember, that's one of those things we talked about in our very first lecture. Seems like it was a long time ago, but just a couple of weeks. So that's how they measure them, in basically large and small subunits, sort of like we have our hands clapped together. So here are our sites, and again, we've got our messenger RNA binding site. It's just a groove that message is going to slide through. And even though the names are not necessarily the same, the A site, again, for arrival, bringing in the new transfer RNA, the peptide site, where you're going to link those 2 amino acids together, and then the E site, that's the empty one, the exit site, where that transfer RNA without an amino acid gets ejected. It's almost like it's spring loaded to spit that empty transfer RNA out.
So is it the arrival site or the amino acid?
Well, it's actually this amino acyl, and that's how the amino acid is linked to the actual transfer RNA. But to help me remember, A is arrival. But that's the official name. Okay. Again, after our discussion and after looking at the video, we're kind of stepping through this quickly, so I'm going to depend on you to ask questions if you have them. Our ribosomal RNA makes the ribosome. Messenger RNA has the code. Transfer RNA, it's this looped piece of RNA that again is going to have some important sites, not the least of which is our anti-codon site, which we already know is going to be a match for the codon in our messenger RNA. But then we also have another site which is going to bind our amino acid. And this amino acid binding to our transfer RNA is via a process called aminoacylation. And so that's where they get their amino acyl site, because it's the transfer RNA with the amino acid attached that comes in together. So how do we get amino acids and transfer RNAs together? And that's with an enzyme called aminoacyl tRNA synthase. This is going to require energy. Here's our ATP. ATP comes in, and we can see that our ATP binds. We actually pop off 2 of our phosphates, as now our AMP stays bound to the synthase enzyme, but that's going to activate the enzyme in such a way that it anchors the amino acid in place, and now the transfer RNA for that specific amino acid comes in, and this enzyme then, using the energy and the change in conformation of the AMP, it links through that aminoacyl bond. And once you link the amino acid to its respective transfer RNA, it gets ejected, the enzyme is released, and you can do the process again. Now, this guy's going around looking for a ribosome with a messenger RNA to be assembled in the right place. So again, energy and energy consumption in our body. We can see how muscles use energy. We can understand how in neurons we use energy for our sodium-potassium pump. But we also have to use energy to make our proteins. And that energy is used in the attachment of our amino acid to our transfer RNA, and there is that ATP molecule. Questions? Now, this is not the same enzyme for all transfer RNAs and all amino acids. You're going to have this aminoacyl tRNA synthase that's specific for the amino acids. And for those that have the 2 different sets of codons, you're going to have 2 different ones for those. Most amino acids will require 1. Those 3 are going to have 2 of these synthases with which they can attach an amino acid to a transfer RNA. Okay? That's how you're going to get the specificity of linking the correct amino acid with the correct transfer RNA. It's not a one-stop shop. Got some kind of puzzled looks. Are we okay? Question. Now's the time to ask the questions.
We're gonna have to know what that enzyme is.
Yes. Now, you just know aminoacyl tRNA synthase. All right? Aminoacyl being the bond. That's the hard. I'm sorry, synthetase. You're correct. Yeah, synthetase. Okay, we good? So just an illustration. This is typically how we've mapped out our transfer RNA. Here's our amino acid. There is the bond that's going to be formed during that aminoacylation step. And then this is just a space-filling model of what scientists think the transfer RNA actually looks like in space. with the anticodon being toward the bottom, and then the amino acid attachment point being sort of over here on the end, not just at the top and bottom like you typically draw them. Okay, but essentially they're the two separate poles of our transfer RNA. All right, so when we looked at our chart yesterday, there were a number, and I think most, well, not the majority, but we had more amino acids in the group that had the N at the end of their codon than any others. And remember, the N meant you could use any of the 4 nucleotides in that third sequence of the codon. So here, for instance, we've got proline. Proline is going to be CCU, CCC, CCA, or CCG. So the codon you remember for proline would be CC and the letter N. N meaning any of the 4 will work. So does that mean, however, that we have 4 amino acyl tRNA synthetase enzymes for proline? No. There's only one. And that one synthetase enzyme can recognize all 4 of those amino acids. All 4 of those codons. Why is that important? Because you only have one transfer RNA that can actually recognize 4 different codons. That doesn't make sense, does it? Because we've always done ATCG, right? Now we throw a U in, right? So that's our 5th one. But U takes the place of a T. Well, here comes number 6. We have a 6th nucleotide-ish molecule. And it's only found in that 3rd position of the anticodon of transfer RNA. And that is called inosine. Inosine is going to be in that 3rd position of many of our transfer RNAs, and inosine is going to allow for the pairing of all 4. Now, it says UCA, but really, inosine is going to allow for the pairing of any 4. So, if we're looking at proline, what is our codon there for proline? Give me one C, U. All right. So if this now is our transfer RNA for proline, and we've got to build our anticodon, what is going to be the anticodon for our C? For C. G, here's a C, G, and for our U, no. I. And with that I in place, that means this N, it doesn't matter. Inosine can recognize whether this is a, U, G, or a C. It doesn't care. And with inosine, when it's present in the anticodon, and that ability to recognize that 3rd position as any nucleotide is referred to as the wobble effect or the wobble hypothesis. So we've got 64 possible genetic codes, 64 different codons we can make, and only 20 amino acids. Well, that doesn't mean we have 64 different transfer RNAs. Yeah, 3 of them are stop codons, so you could say 61. No, we don't have 61 transfers because with this wobble effect, all of our, all of our transfer RNAs that can recognize N, it's just one transfer RNA that can recognize 4 possible codons. Brody, still kind of?
Yeah, so,
I's a wild card.
right.
I'm uno. You got a wild card. It doesn't matter what's there.
So basically, like if we had like CCI, then we would recognize 4. If we had,
Now remember, this I is only in the transfer. It's only going to be in.
it's only going to be attached, or it's only going to be there when you see the N.
Correct. This is our codon. This is our messenger RNA. This is our transfer RNA. Okay? That I is never going to exist in messenger RNA, ribosomal RNA, not to my knowledge.
When you have the N, which means any of them,
Right.
the U, C, the A, or G, then you're going to have the I.
Correct.
But if it said like CCA, then you wouldn't have I.
Exactly. Then it's specific. Because remember with our methionine, UAG, well, we just had one codon, one anticodon. So we just have one transfer RNA methionine, and it only recognizes the one. Because number one, it's the start codon. We have to make sure it's in the right place. Right? So wobble, we get the idea of the wobble and how that basically is facilitated. Of the wobble? The wobble is referring to the third position of our codon, which really relates more to the third position of our anticodon in transfer RNA. Where you,
correlation between inosine and wobble.
yeah,
Good.
yeah.
And it's only in transfer RNA.
Inosine's only in the third position of our transfer RNA. It's not in the first, it's not in the second, it's only in that third position. That basically says, I don't care what you put there, I'm going to bind to you. As long as you got the first two right, then everything else doesn't matter.
So, since it's wobbly, does that mean it's less stable?
No, no. Now remember, that the interaction that's binding, that fit, man, it's quick. It's not going to exist long. Because that ribosome's zipping along the mRNA, and you're putting those peptide bonds together, assembling the amino acids in a hurry. Okay. So when we look at our message, we're going to have, again, those upstream regions and our downstream regions. When we say upstream, downstream, we'll say upstream from our start codon from our AUG. So this is our messenger RNA. We've got our ribosomal binding site. Remember, ribosome takes about a 30 base pair footprint. Remember, we've got sort of a pre-messenger RNA, then we modify it. And in eukaryotic cells, we're going to have the 5' cap. What is used to 5' cap? What chemical modification? We're replacing a hydrogen with a methyl group, so it's a methylated. It's a 5' methylation that protects the 5' end. And then here at the 3' end, we've got our poly A tail. Maybe upwards of 200, 250 A's, just A. So if any degradation is going to happen, it's got a lot of A's to chew through before it's going to get to the important stuff. And situated between this untranslated region, which is capped, and our ribosome binding site, we've got our coding region with our start AUG and one of our 3 stop codons with which to build our protein. All right, so initiation. Again, we said we're going to have the small subunit, we're going to have our messenger RNA, and we're going to have the initiator transfer RNA. Which one is that always going to be? Methionine, always. So we've got our AUG start sequence, and we've got our UAC anticodon of the transfer RNA attached to methionine. So here's our small subunit. That small ribosomal subunit is going to bind to several proteins that are called initiation factors. What else is going to be bound? GTP. You have to have energy. Not just to assemble the amino acids on the transfer RNAs, but now we've got to have energy in the form of GTP to put all this thing together and assemble it. So we bring in our mRNA. We've got our now initiation factors and GTP bound to the small subunit. There's our methionine transfer RNA. That all gets stuck together. And notice that methionine and bound to the start codon, it's starting in the P site of our small subunit. Not in the A site. This is the only time you'll see that. And once we have several of our initiation factors leave, but the one bound to GTP is in the E site. Once we have that configuration, now we put the lid on it. We cleave the GDP and the phosphate so we release energy. That is essentially like kick-starting the whole process. You have to cleave the GTP for this thing to get started and start the elongation process. And really the energy to secure the two subunits together and hold it in place. So there we have in our P site, there's our methionine. Now we're ready to go. And we're ready then for the elongation step, which is in fact bringing in the next transfer RNA with its amino acid into the A, or the arrival site. Has everyone got initiation? Got to have energy. You've got to have initiation factor proteins. You got to have the subunits, and you got to have the mRNA, and you got to have your transfer methionine. Those are the players involved in the initiation. Now, this is wordy. But as we saw in the video, we understand the significance of the A, the P, and the E site, right? Anybody unclear about how that's working? Everybody clear about how the growing chain is moved from the tRNA of the P site over to the amino acid on the A site as it slides? We saw that in the video. What happens to that empty tRNA when it moves into the E site? It gets ejected. All right, so that's really all that we have talked about here in this really, really wordy slide. For those of you that like words and likes instructions and steps, this is for you. And this is for the picture folks. Because we're also going to use proteins and energy in the elongation step. So here's initiation coming in, okay? That's just that we're going to start at the top. This is what we built. So now we've got our second amino acid in our sequence bound to its transfer RNA. So it already visited that amino acyl tRNA synthetase. All right, it got linked up. Now we have these molecules called elongation factors. And elongation factors plus GTP, so energy, bind to our amino acylated tRNA to bring it into the A site. The transfer RNA with its amino acid alone is not just going to get there. It has to be brought in with our elongation factor and energy. And so once we have it in place, we cleave the energy. The elongation factor pops off, and now we have our tRNA number 2 with its amino acid number 2 in the A pocket. Once we're together, we're going to form a peptide bond, and the peptide bond basically is going to be formed as you pass that amino acid from the first P site over to the A site. Everybody see that? So now we have our peptide bond formed, so we can look at it like binding of the next tRNA. Step 2, peptide bond formation. Step 3 is translocation, so the actual moving. This is going to be another elongation factor. This elongation factor comes in with GTP. As it comes in, it's going to cleave off a phosphate. You can see it's actually also bound in this A site. So what this elongation factor G, I like to say G for go, elongation factor G comes in and it wants to move into that A site area, so it kicks the transfer RNAs over one spot. And because of the energy stored and the energy released, it's able to slide the ribosome or slide the messenger RNA, however you want to look at it. That energy causes the ribosome to move. Now we've got our growing chain and the transfer RNA in the P site, the empty first methionine transfer RNAs in the E site, and it gets ejected. Once the ejection happens, once our elongation factor G is in the A site, it pops off and does it again. Now what we have is we have another round where we've got a transfer RNA now with 2 amino acids in the middle, and it just keeps doing this and doing this and doing this, and that's the elongation.
So basically, the elongation factor is just making it longer.
Exactly. You've got to have one to bring in the transfer RNA, that's the T. You've got to have the T elongation factor with energy, and you've got to have the G elongation factor to make it go, to make it slide. For each cycle that comes around. Are we good? So again, we started real simple with that video. But now if we hadn't seen that overview and everything in motion, it would kind of get really muddy. But now as we're just adding the finer and finer details, hopefully we're all kind of hanging in there, it's making sense. So initiation, elongation, now we've got to get to the termination part. And that's going to be our termination factor or release factors. And again, these are proteins, look like transfer RNA. They have what amounts to an anticodon that can recognize, an anticodon for our stop codons. And when they come in bringing that energy and they bind to a stop codon and they end up in that A site, that energy then is used to just blow the whole thing apart. It blows the subunits apart, the messenger RNA is released, the tRNAs are gone, and the growing protein is finished and released. Guess what? That's translation. That's it. We're done. So in addition to our players, our RNAs, the amino acid, also remember the initiation factors that we need to assemble it with GTP, the elongation factors that we need to bring in the transfer RNA with its amino acid, the elongation factor we need to move the ribosome along, again with GTP in both, and then we have our release factors that also have to have GTP. You see the common theme here? GTP energy at each and every step. Elongation takes two of them. We also have to have energy to link the amino acid to the tRNA, but that's the only place we see ATP instead of the GTP. All right? Now, whenever we looked at transcription, we said that in prokaryotic cells there were multiple codes for different genes within one messenger RNA. Do you remember what term we applied to that? It was poly something. Polycistronic was the term for that. In eukaryotic mRNA, we're only going to have one gene, which is going to encode for one protein. So does that mean in our monocistronic eukaryotic cells, there's only ever going to be one ribosome attached to that one message at a time? The answer is no. Because what you can do as the first ribosome gets started and moves far enough out of the way, another ribosome can come and bind. And it can start making the protein, and again and again. So we refer to this as polysomes. I mean, and just this line of ribosomes just sliding down, making protein after protein and keeping almost this assembly line going. You can really maximize how much protein gets made from one single message. You can do it quickly before the message is degraded or that message deteriorates. So you don't have to wait for one complete protein to be made before you start the process again. And often when you look with an electron microscope, you can see these ribosomes lined up out just in the cytoplasm. So that's what we mean by a polyribosome, just several on the same message at one time making the proteins. So when we use our transfer RNA and our ribosomes to assemble the amino acids in the right sequence, what level of organization are we talking about? Primary. Just the right sequence of amino acids. That's the primary structure of a protein. But we have to make sure that protein gets folded into our secondary structure and our tertiary structure, and that then gets assembled in our quaternary structure. So protein folding is extremely important And so we have these proteins that hang around that often assist after the proteins are coming out of the ribosome, sort of like a pasta machine. Even before it's released, our chaperones are going to bind to those amino acids, and they're going to start the process of folding a protein.
Because in that video it said that it just started to like make a structure.
Well, and again, that's a very simplified version
So the
of the video.
chaperones are the things that actually.
They're going to have, now some can self-assemble, some can self-fold and begin certain levels of that process.
How would we know which can do what?
Uh, well, until you know the protein that we're talking about, you can't just assume it's all one way or all the other way,
So
which is
like,
which.
we need to know like, for the exam, like if it's this protein, it would have a
No, no. Now, we mentioned frame shift mutations when we were talking about the production of our mRNA. We talked about the addition of 1 or 2 or 3 nucleotides or the deletion of 1 or 2 or 3 nucleotides, which would cause a reading frame of our codons to be moved. Well, the result of those mutations in our messenger RNA can yield proteins that may be significantly different depending on what amino acid sequence are now encoded based on those mutations. So we've talked about the frame shifts. Yeah, that's the insertions, deletions. That's our foundation on which we're building. But with our frame shift mutations, if that change does not alter the amino acid sequence, it might be in that third position. So if you change that third position of our codon and that amino acid that it encoded for had that wobble, it doesn't matter what that third position nucleotide is, it's going to be the same amino acid. You understand that? So in that case, if the amino acid doesn't change, even though you change the sequence of the messenger RNA, that's called a silent mutation, because the protein's going to be perfectly fine. If, because of our mutation, we change a single amino acid, and again, we're not talking about insertions, deletions here, we're just talking about a change. This is going to be a change in either the first or the second codon. If we change a single amino acid, that is a missense mutation. Sometimes one amino acid difference is going to completely change the function and the structure of that protein. And again, sickle cell anemia is one amino acid change in the hemoglobin gene. And you see what kind of effects that can have. Now, the last mutation, which can be changes, insertions, deletions, if you have a premature stop codon, that is called a nonsense mutation. So I know that a lot of these missense, nonsense, frame shift, kind of sounding the same, but it's important to keep those categories of mutations apart to know which is which, because there's a big difference between a premature stop and a single amino acid difference. Okay?
So on the nonsense mutation,
Correct.
it's got the premature stop time, so it just stops the whole thing.
Yes.
Now, why does it not stop the whole thing in frame shift if we've got the insertions or deletions, but it does it in nonsense?
Okay, if you have a frame shift, if you insert one or delete one and you change the reading frame, but that reading frame, those codons, don't encode for a premature stop. They just change amino acids. Instead of a proline, you got an alanine. Instead of a glycine, you got an isoleucine. Instead of a valine, you got, and you keep going until you finally get to a stop. That's not a nonsense mutation. But this is if you're making the protein and because of a single change, instead of having a different amino acid now, halfway through the protein, instead of valine, you got to stop. That's a nonsense. Okay. Everybody got that?
So we're saying if we have to stop, it's a nonsense.
Correct.
And the stop could be caused by either a substitution, insertion, or deletion.
Got it. Couldn't have said it better myself. In fact, I'm not going to. So again, here's our example. missense mutation instead of a glutamic acid because of our one change in the messenger RNA. Now we're going to get a valine. Nonsense, we expect our protein to have leucine, but one nucleotide change, and there's our premature stop. And then a silent mutation, notice proline. You remember our proline over here, CCN? Well, our mutation happened in the third codon, but we still get proline. So that's a silent mutation because it didn't change the amino acid. Yes, ma'am.
How often do mutations actually occur?
Whew. Some genes are more predisposed to mutations than others. Your body, because of the proofreading nature of our polymerases, and because of genes that are called, and proteins are called tumor suppressor genes and proteins, your body's really good at correcting mistakes. It's not perfect, and that's how people end up with cancer, because we're continually bombarding ourselves with environmental agents and carcinogens and everything else. It seems like every day something else causes cancer. You can do the best you can, but as your cells divide, invariably, there's going to be a slip up somewhere. So you would have to know what particular gene we're talking about. You would have to, probably know the age of the individual. There's going to be a ton of factors that are going to affect mutation rates.
So it could happen commonly, but your body can catch it and just destroy the cell if it needs.
Correct. And, and depending on the genes, there's going to be a gender bias. Sometimes you're going to have more mutations in a particular gene in females. Sometimes it's going to happen more commonly in males. There's going to be the ethnicity. Your genetic composition from a heritage standpoint could make you more predisposed or less predisposed to certain mutations in certain genes, and that leads to an increased risk of certain diseases and or cancer. So that's a real long way of me saying I don't know. But there's just a lot of factors that can affect the predisposition of a gene to mutation. And sometimes it's just happenstance in a lottery. You get the right mutation in, let's say, one of those regulatory genes that control cell division, those checkpoints between G1 and S and G2 and M, and all of a sudden you don't have those brakes on anymore. There you go, you got a tumor. So now as we get into our protein, the chaperones, they help fold the proteins. But there are also instances where we have to really alter the protein. And one way we do that is cut the protein up. And so some of our protein sequences are only there to help the important parts of the protein get folded in the right place. And as you look at this example, the orange piece, it was there so that we have this loop on the right side and this elongated part on the left get in the right place so you can link these disulfide chains together. And then that piece is cut out in the middle, leaving you a functional dimer, a protein that consists of these two chains, each being folded in the correct place. Now, in many instances, even though methionine is the first in all, a lot of times the methionine is cut out. So that's removed. That's just hey, we get started here. Whether we need it or not, that's to be decided later. But we also have a sequence that's called a signal sequence. It's in the first part of a protein. So as a ribosome is beginning to produce this protein, that signal sequence, as it's coming out, it's going to bind to receptors on guess which organelle, if it's going to be a secreted or a transmembrane protein, the endoplasmic reticulum. And it's going to be that signal sequence that causes that ribosome to stick to the ER. So when you look at rough endoplasmic reticulum, these are not ribosomes that are permanently housed and stationed on the ER. These are ribosomes that bound to a messenger RNA, started making the protein, and since the protein had this signal that said, I need to be secreted or transmembrane, that's what brought the ribosome in to finish the process of forming the protein. But that signal sequence only gets it there. You cut that signal sequence off to yield a final mature protein. Now, much the same that we modify our nucleotides. We can methylate our nucleotides. We put poly-A tails. Chemical modifications to proteins. You can put a methyl group. You can add a phosphate. You can put an acetyl group, and certainly the addition of sugars, which starts in the ER, but you really have the bulk of this sugar addition glycosylation in which organelle that also functions as packaging and shipping? The Golgi apparatus. The Golgi is where this is going to mostly take place, even though it begins in the endoplasmic reticulum. Now, we're familiar already with those segments of DNA that really don't do anything. They're only there as a buffer for mutations. A lot of people call it junk DNA. And it's that part that in our message we have to splice out and then glue the good parts together. What do you call the parts that you splice out of the messenger RNA? They're in the way. Introns. Introns is that stuff that's in the way. You got to splice them out and glue the exons, which are the good stuff, together. Well, proteins also have junk pieces that may have been responsible for folding or other binding functions of the premature protein. Those have to be spliced out so you can yield a final productive protein. So in much the same way that we have introns and exons in our RNA, we have inteins and exteins in our protein. And so here you can see a precursor. We need to link the green and the purple together so the yellow portion has a secondary structure sufficient to bring these two pieces close enough together that an enzyme can cut off the yellow piece and stick the green and the purple together. Here we have a couple of two different proteins forming a quaternary structure. They have their two inteins that allow these to bind close enough together. You see how that brought those two pieces, the red and the I guess purple, close together. Enzyme comes in, cuts out the inteins, sticks them together. So even when we start making that polypeptide chain, we haven't arrived yet at a fully functional mature protein. There's still a lot of post-translational modification that goes on. Now pay attention to those names because we had post-transcriptional modification. That changes the message. Remember we 5' methylate, we add the poly-A tail. Post-translational modification is the cleavage, the phosphorylation, the methylation, the acylation, the splicing out of our inteins. All of that happens after the process of translation. So as we are making that protein, I've already introduced the fact of the signal sequence that binds that growing protein to the endoplasmic reticulum. That's the way that we're going to begin to target where these newly formed proteins are going to go. So we can have just our free-floating ribosomes out in the cytosol. We can have our ribosomes associated with the endoplasmic reticulum. But then we're also going to have these other organelles that are going to have materials provided by your nuclear genome. That's where the code is, and those proteins have to be made and somehow shipped to these targets. So again, when we look at how that happens, well, if you're a cytosolic protein, you're just going to be made on a free-floating ribosome. Protein synthesized, chaperones fold it, it gets processed, and then it just falls off and stays. And that's the easy one, right? But if we're going to other organelles, we're going to see that they are, again, associated with free ribosomes in the cytosol. But we're going to see that that protein is going to have a signal, a targeting signal in its sequence that's going to target it to a particular organelle for import. So again, it's going to be that amino acid targeting sequence that's going to direct us. Okay, are we supposed to go to a mitochondria now? Are we supposed to go to a chloroplast? Now, if this is going to be a free-floating one that's going to be transported into the lumen of a particular organelle, we're still on those free ribosomes. But if we are going to be secreted or if we're going to be transmembrane, then we're going to have to go into the ER. And again, right after you begin to make that short peptide, there's going to be a signal sequence. And that sequence of amino acids is what links the ribosomes to the ER. And as it's being extruded into the lumen of the endoplasmic reticulum, there's an enzyme just inside that membrane. It's like a pair of scissors that snips off that signal sequence, and that protein just continues to get produced and extruded into the lumen of our ER. So here is an example of how this might work. Here we are out in the cytoplasm. We have initiated translation. We've assembled our protein subunits, our ribosomal subunits. There's our single transfer RNA in a growing polypeptide chain. And so we have this signal sequence binding protein. And this signal sequence binding protein binds to that first leader signal sequence of our protein. That then causes this whole unit to bind to the receptor on the endoplasmic reticulum membrane. That's what's holding the ribosome in place. Now, here you see this little sharp-pointed thing in orange, signal peptidase. That's going to be like our pair of scissors. So as we continue making the protein, and you can see now it's starting to extrude through our protein channel. It's coming on in, and as it gets further in, there we've cut off that signal sequence. The protein continues to get made until we finally completed the process of translation, and now we have this particular protein that is free-floating inside the lumen of our endoplasmic reticulum. Now, once you're free-floating in the ER, what might your target destinations be inside the cell? What would be the most likely targets? Because think about it, you're inside the ER. So you potentially could be a hormone that is going to form a transport vesicle off of the ER, go to the Golgi, get modified in the Golgi, get into a transport vesicle from the Golgi, fuse with the plasma membrane, and then with a process of secreting that, what process do we call that? Getting stuff, exocytosis, that vesicle fuses with the membrane, and now all the stuff that's inside the ER, that's going to be secreted out. Now, if that vesicle happens to be a lysosome, we might be making an enzyme that then gets transported and is just floating inside the cytoplasm in a lysosomal vesicle, waiting to fuse with a phagosome to process and digest materials. Now, at the same time, if you have a hydrophobic region, remember those hydrophobic amino acids at the transmembrane portion? If you have a hydrophobic region, it's going to get stuck in the middle here. So you'll have the part that extends down into the lumen of the ER. You'll have a part that gets stuck in the ER membrane, and then you'll have a part that's still sticking out here in the cytoplasm. So when that then fuses with a membrane, maybe the membrane of a mitochondria or the plasma membrane, what's on the inside of the ER? That's going to be equivalent to the extracellular domain. What's left here on the inside, the cytoplasmic portion, that's still going to be cytoplasmic portion, but now of a transmembrane receptor or channel on the plasma membrane. Do you see how it sort of turns itself inside out? So it's almost upside down what we think of with the ER versus what we're going to see as a transmembrane protein. So in the ER, we got a situation where we are going to look at the folding of our proteins. And the folding's not always going to be correct. And so we have enzymes that are present inside the ER that are going to say, okay, you didn't fold right. We're going to chew you up. It's like a paper shredder. And so we're going to chew them up. Many of our proteins are joined together into a secondary structure and even the tertiary structure with disulfide bonds, not the least of which are going to be our antibodies. And those disulfide bonds are formed inside the ER with disulfide isomerase enzymes. So we're going to get, even that level of tertiary structure folding inside the ER. So there's a lot of modification that goes on in the ER. And the other one that we didn't put on this slide, again, was the beginning of glycosylation. But honestly, I really would rather you associate glycosylation with the Golgi, because that's by and large where that's going to happen to the largest degree for our proteins.